This is a recap of the breast cancer cell line activity. I've already saved it as a different name and I've loaded the data. Here we want to remove a column from the expression data frame. The first column are gene symbols. First column contains gene symbols, and we want to remove that and make them into the row names. We use a -1 to get rid of the first column, and the feature we want here is for symbol. Now we have our gene expression matrix, and it looks exactly as we were hoping. So you can put some commands there. Now let's look at the cell speed data frame. We'll look at some of this information. If we want just the column names, we use the call names function. Great. This unique function will give us the unique values that occur in columns 2 and 3, the correspondence between cell line and the cancers from which they were taken. To get information on the experimental conditions, we make a table, and we see a number of different conditions. We're going to use the hyaluronic acid collagen substrate. And this image shows us the extracellular matrix for the cell, which is in red, and hyaluronic acid and collagen are some of the molecules that are in the extracellular matrix. So this is the region in which the cells are supported and move, and indeed our experimental condition is biologically significant. Now we have a data frame with just the information on the hyaluronic acid condition. We want to reduce this further by pulling out those cell lines with a breast cancer diagnosis. Okay. So we want to extract these samples from the gene expression matrix. To do the log transform of our expression matrix, we add 1 here. This is in case there are 0 values. When we take the log of 0, we get an infinity. So this is taken care of. Again, we can click on the object in our environment tab to make sure everything looks good. Here we're going to pull out the column numbers for the experiments that we're interested in. It's columns 44 and 58. So we have those numbers in this new object called experiments. So we can pull those columns out of our expression matrix and we'll label them slow and fast for the 2 different cell lines. Okay. Now to make a differential gene expression feature, we'll subtract the values for the fast cell line, which is in column 2. We'll subtract the slow gene expression values, create this new object, DGE, and make a new matrix by using the function cbind to add that feature as a 3rd column. And you're asked to make a histogram, and we should put a title and appropriate axis labels. Now we want to know which genes have a differential expression greater than 5 and those with a differential expression of less than 5, because those are the genes that are highly expressed, more highly expressed in one condition versus the other. So we're using a logical, the DGE less than 5. So 94 other genes in the left tail of the histogram to do the same for genes in the right tail of the histogram. We can delete this information and now put in a greater than 5 logical condition. Right. So now we're going to order the differential gene expression values from higher to lower. And once we have those, the rows that correspond to the differential gene expressions values that we want. We can put that object here in the row section for our matrix. And let's see, what did I do? Oh, all right, I put the wrong object in here. I'll put DGE instead of the ordered. All right, so now we've reordered the matrix, so we have this third column, and the rows are ordered so that we have the highest expression in the fast line over the cell line in the third column. To look at what's at the bottom of that matrix, we can use the tail function. Okay. You can load this image and read the text around here to find the impact of removing some of the genes on this particular cell lines. Okay. Now we're writing the row names for the top 50 genes in our ordered matrix. That should give us a new file, and we can open it, and we see that these are the genes that we had in our activity, and now we have the top 50 instead of the top 15. And we're asked to go to this server, which uses a large language model to look for functional themes in our genes. Copy them, put them in the query window, and run. And our genes are over-enriched for functions that are related to extracellular matrix remodeling and cell migration.